Welcome everybody to our webinar today. I would like to begin by acknowledging the traditional custodians of the land on which we meet and pay respect to their elders past and present. We extend the respect to Aboriginal and Torres Strait Islander people here today. I'm on the Wurundjeri land of the Kulin Nation, and I hope between all of us we do cover most of the country today. Just some housekeeping. Please ensure that your microphone's on mute. Raise your hand to speak in the Q&A session, and please use the chat function for any questions. We will have some time to answer them. And please send questions. We often do not get many questions. We'll make a recording available to watch on the website and YouTube channel. And we would also appreciate your completing a post-webinar survey after the presentation. So just to say what we're doing today, members of a DSMB are expected to evaluate accumulating data to monitor study conduct more and more. So for that reason, ACTA is hosting a 3-part webinar series to provide education on the complexities of serving on a DSMB, including interim stopping rules and the challenges of stopping trials and safety monitoring in adaptive trials. We're doing this today through a series of 3 webinars. Today is the first one of these. And then we'll have one in 2 weeks' time with the same speaker. And then we'll have one in October by Tom Snelling and Julie Marsh, where we'll talk about adaptive designs. Sorry, the next one we'll have in September will be about the challenges of stopping trials for benefit. And Lehana will share an example with us. I won't go over exactly what we will be covering in today's session, but I'll just mention the title. So we'll look at interim data sharing by data safety monitoring boards, what to share with whom and why. And then it's my absolute pleasure to introduce our speaker, Dr. Lehana Tabane, today. He is from Canada, where he's a professor of biostatistics at McMaster University and a fellow of various institutes throughout Canada, America, and South Africa. He is a past president of the Society for Clinical Trials, and he has received many recognitions for his work. He's also a prolific co-author with many methodological and applied papers, and has a legacy of mentoring many students and junior faculty. I also want to share some more personal introduction. Lehana and I met many years ago when he was instrumental in setting up biostatistical training in sub-Saharan Africa. At that point in time, there were no dedicated biostatistics degrees. There were stats degrees and there were biostatistics courses, but not a specific degree. Now there's at least one degree in sub-Saharan Africa where Lehana played a big role. There might be more that I'm not aware of. And what I admire most was the very applied roles he has played in research institutes, where he straddles rigorous stats methods with applied need to get information to help patients. He's a distinguished biostatistician who has held senior leadership roles that go beyond the traditional scope of a statistician. For example, he served as the Vice President of Research at St. John's Healthcare, Hamilton, and Scientific Director of his Research Institute. These key kinds of roles are typically held by clinicians rather than statisticians. So I think we're inspired that his career shows us that statisticians can also step into these leadership roles and not just be stuck in the methodology, but influence strategy and policy. So Lehana, thank you very much for your time. We do appreciate it.
Thanks, Aneka. That was a very kind, generous introduction. We have been struggling with the slides, but I've sent my slides to Natalie, who's going to share the slides, and we'll try and make this work somehow.
That looks good.
All right. I hope everybody can see this. If you're struggling with the slides somehow, please use the chat. Use the chat option, and Natalie or somebody will monitor that. We'll try and make things happen. Yeah, next slide, please. Now, this is also typical of what we try to do in Canada. I'm outside of the country now, but whenever I'm in the country, we will also go through a land acknowledgement where we would acknowledge that McMaster or St. Joseph's Healthcare Hamilton, which is where I also work, recognize the lands on which we land and provide care, which are the territories of the Mississauga and the Haudenosaunee peoples. And for thousands of years, the first people sought to steward precious resources and share the land with others. In the territories where I work, the subject of the Dish with One Spoon Wampum Belt Covenant. Next, please. And you can go next again. And Dish represents the shared land, while One Spoon reinforces the idea of peacefully sharing and caring for the lands around the Great Lakes in the spirit of mutual collaboration. McMaster and St. Joe's, where we work, we pledge to continue to work together with the Indigenous peoples in building a more just society where their gifts and those of other people are nurtured and honored. Next slide, please. So I'm actually outside of the country. I'm calling from Addis Ababa, Ethiopia. I started the journey in Kigali a week ago, where I was meeting with a group of investigators around the continent who are involved in the REVIVE trial. And the REVIVE trial is an HIV clinical trial, and I chair the DSMB, and is trying to investigate whether using azithromycin could actually help to perhaps prophylactically prevent infections of those people who are put on ARTs. And so I chair the DSMB, and the investigators meet annually, and this year they were meeting in Rwanda. So it's been quite a journey, and I didn't actually realize that I was on a different time zone now. I forgot I had committed to doing this talk, and it almost failed. So it's 3 o'clock in the morning. So if I'm sounding a little bit with a heavy voice, it's because I'm so terribly jet lagged. Next slide, please. I don't have any conflicts of interest to declare, but I am a professor. Like everybody else who's a professor, I do get credit for giving presentations. Next slide, please. So really the key message is to discuss the potential merits and challenges of an increasingly common practice of data sharing, in particular sharing interim combined event rate, and I'll explain what this is. But I also first start with the fundamentals of about monitoring clinical trials. Next slide, please. So what is a data safety monitoring board? Yeah, you can go ahead, do one more. Good. So it's a committee of about 3, sometimes it's 15, sometimes it's 5, sometimes it's 10. But they're individuals really with expertise in medicine, ethics, biostatistics, or in general in clinical trials. And it comes with different names depending on where you are. In Canada, we often use the Data Safety Monitoring Board and the acronym DSMB. But in other areas, in other textbooks or where people have written about this, they often use Data Monitoring Committee. So I would be using these two acronyms or terms synonymously. I just wanted to make you aware that it actually does come in different names. Next slide, please. All right, why don't you do all the responsibilities and then we can get. All right, so the key responsibilities here may vary from trial to trial, and they may include really just providing the, providing the committee with information on harms and safety, and all they do really is to monitor safety in a trial without necessarily being charged with looking at both safety and efficacy, although traditionally they tend to also look at both, or to identify emerging problems with the conduct of a trial without necessarily being worried about certain things regarding outcomes. And I'll come to this issue as we go through. And they could be monitoring things like recruitment, randomization problems. And typically it's because the investigators may be worried about the integrity of the trial. Sometimes it's a long trial and they anticipate that they could have issues with designs, and they would like some group of colleagues who are external to the trial to monitor the integrity of the trial to help them. Sometimes their job is to look at the futility because investigators are worried, or they may be asked to monitor feasibility of the trial as it's being currently designed. Whatever happens, really, one of the key goals is to be able to see whether along the way the trial has come to a point where we can actually feel comfortable that they have met the objectives of the trial, and therefore they might actually even recommend stopping the trial early. And we'll talk about the issues related to that. So interim look of accumulating data in the trial, it actually started in the 60s. Next slide, please. Right. So this is one of our colleagues, Susan Ellenbeck, who is well known in data monitoring and has written quite a number of books about this. And Ellen really has a fantastic presentation, really giving the history of where it all began in the 60s. So NIH used to have this major trials, and then they would actually, and mostly it was about cardiovascular trials or diabetes trials. And what happened was that they would then have interim looks at the data, but this was actually done by the investigators themselves, right? But at some point they realized that perhaps this is not the best way to do this. And so they would ask a group of people outside of the trial to actually join them to be able to do this. So it wasn't always the way it is today. It used to be done. And when they did multiple looks, they didn't even adjust for anything, to be honest. Next slide. And then what happened was that they realized that this practice of the investigators themselves doing it was not really optimal. So they convened a project which was led by Greenback, and Greenback actually produced a report of how perhaps to best do this. And this is a report that was known as the Greenback Report, and you can find it online. It was published in 1988, and it really provided some impetus of how to best approach monitoring, accumulating data in a trial. At the time, it was not even called the Data Safety Monitoring Board or the Data Monitoring Committee. It was called a policy board. But this really was the beginning of perhaps a much more formal process of doing this. Next slide. So the policy board, the idea was they would have interim review of the process, would be optimized, they would minimize bias because it would be done by a group of external colleagues, and this would enhance the objectivity. And the US government started now using DSMBs on a regular basis for NIH trials. And we'll see that even the FDA trials started actually using the process. Next slide, please. So who appoints these people, right? For most trials which are done by the Canadian Institute of Clinical Trials is the steering committee or the PI. If it's PCORI trials, it's patient-centered orientation research initiative, which this is US, it's also the investigator who would actually appoint them. NIH, same thing. I think in Australia, probably the practice would be the same, that if it's a federal agency funded trial, then the PI or the steering committee would appoint the board. If it's a pharmaceutically sponsored trial, typically the sponsor or the pharmaceutical sponsor would be the one appointing people to serve on the DSMB. The Veterans Affairs Group, which are trials funded by the Veterans Affairs in the US, they would actually be the ones appointing members of the DSMB. Next slide. The primary mandate is to safeguard the interests of the trial participants. The idea is to preserve the integrity of the trial and the credibility, and perhaps to allow an external group of people to be able to provide with timely and reliable insights without necessarily jeopardizing that credibility or integrity of the trial. Next slide. All right, so the way this would actually be done is there would be a DSMB charter, and this provides, the standard operating procedures of how the DSMB will function and how they will maintain confidentiality. and how the interim data would be interpreted. There would be some guiding principles about how to interpret the results. So as a result of that, the DSMB should have clinical trial experience so they could actually do this job successfully. And they have sole access to interim data, perhaps assisted by what we would call a DSMB statistician, who is a statistician who works with the team, but they're dedicated primarily to supporting the DSMB in their mandate. They would be multidisciplinary in nature, and they would be independent with no significant conflict of interest in the trial, financial, personal, or otherwise. And that would include clinical experts in the disease area or treatments being investigated. And then they would have a very experienced statistician as part of this process. Sometimes we've had patient reps who are knowledgeable in clinical trials to be part of the process. The reason why I showed you earlier on that sometimes we don't have the maximum number determined for a DSM is because if you have an international trial or global trial, sometimes there might be a need to have regional representation in the DSMB, and sometimes I've seen DSMB as high as more than 10. So this could be to allow some regional representation on the DSMB. Next slide, please. All right, conflicts of interest. They could be many. So typically this is important because we want to make sure that the members of the DSMB are independent and therefore have no perceived or real conflicts of interest. This could be financial. It could be, when you have a patient involved and some of these patients, may have their loved ones be involved in the trial and have vested interest. You also want to make sure there's no intellectual or emotional involvement that could actually jeopardize the role of the DSMB members. So sometimes if you have a DSMB being too close to the PI, that could be a problem. Now, could you have regulatory involvement in the DSMB? Typically, that may present a challenge, especially if the regulators themselves may be involved in the decisions about whether a therapy or a treatment is approved or not approved. So it's very rare sometimes to have a regulatory involvement in the DSMB process. Next slide. So what's in the charter? Who serves on the DSMB membership, their primary responsibility, how conflict of interest would be declared and managed, procedures for ensuring confidentiality, the purpose, structure, and frequency of meetings, handling and managing minutes of the meetings. We often have open session for the DSMB meetings and the closed session. And then there would be a statistical monitoring plan if there's some statistical rules that would be used to determine whether or not a trial could be recommended to be stopped early or not. So what would be contained in the open or closed session reports and the process of how recommendations would be communicated to the investigators or the steering committee. Sometimes DSMBs have had to communicate directly with research ethics board or with the sponsors, people who actually provide the funding for a trial. Maybe I should be using the word funders. So that is often also included in the charter. Now, what is often sometimes missing, especially for investigator-initiated trials, is indemnification of DSMB members. I'm highlighting it because I'll say a few things about it. Next slide, please. All right. Who are other non-DSMB members who are involved in trials which may have interest in its progress? So you may have the sponsors, you may have steering committee members, you have principal investigators, you have other study investigators that are not necessarily part of the steering committee, you have those that are funding the study, you have the data analysis center, which may not necessarily be part of the sponsoring institution, maybe a CRO that is doing this job. And then you have primary trial statisticians who are actually interested in this as well. But these people may not necessarily be serving on the DSMB. In fact, you wouldn't want some of them to serve on the DSMB because of potential conflicts of interest. Other non-DSMB members who are not directly involved in the trial but may be interested in the results of whether the trial is on track, Now, the study participants themselves, the press, I'm sure you'll recall with COVID, everybody was interested in the interim results of COVID trials. The public, the Research Ethics Board, regulatory agencies, and advocacy groups, if it's a particular disease that's actually perhaps more prevalent in one particular group. I think with HIV trials at the beginning, the HIV groups were interested in interim results of almost every trial that was being conducted at the time. And other DSMB trials of similar trials could also be interested in what's happening. Next slide, please. Now, so the possible recommendations from a trial that is actually being monitored by a DSMB could be, well, we've looked at the data and we considered all the things that were provided to us. We think we should continue the trial access. Or they may say, well, we recommend that you actually stop the trial early because we think you have met your objectives of showing that the intervention is doing more good than harm. There's overwhelming evidence based on what we've looked at, or there's safety issues that really they're worried that perhaps things are not as they had originally been thought of, and there's worries about increasing harm in one of the arms. Or it could be that they say, you better cut your losses. We don't think you'll ever be able to answer the question. And therefore, the trial is recommended to stop early for futility. Sometimes they may not recommend stopping the trial early, but they may recommend modifying the protocol or certain aspects of it. Next slide, please. All right, so if they say modify the protocol, this could be modify the eligibility criteria, modify the doses and the scheduling, and perhaps you might want to institute some screening procedures to beef up what you have, or change something in the informed consent process because new information has come to light that may impact people's willingness to participate in a trial, that perhaps that information may not have been available at the beginning of the trial. So there's various things that they may ask the investigator to consider modifying. But remember again that whatever comes from the DSMB, they're nothing else but recommendations. They're not orders that people should do this. They actually just do provide recommendations, and the steering committee or the PIs do have final decisions. Next slide, please. All right, possible reasons for stopping early said it could be for benefit, it could be for harm, it could be for futility. Sometimes, you have the DSMB and you tell them, guys, we've run out of money. And they may say, well, because you've run out of money, we don't think the trial is actually meeting its obligations to the patients and we recommend stopping. Or sometimes the data quality could be so poor that honestly, the integrity of the trial is being jeopardized to the extent that the DSMB would recommend stopping the trial. Possible arguments for also stopping early could be scientific. There's external evidence showing that the question has already been answered, given maybe external other trials looking at this, and therefore there's no longer any basis for equipoise for doing that trial. Or it could be the ethics related to that equipoise, resources, like I said, or people have just become tired because it's a trial that have gone on for too long. It's no longer recruiting as much. The investigator no longer have the same enthusiasm they had at the beginning. Or certainly it could be other things, or there could be a fears of legal action of what's happening because perhaps a similar trial or a participant in the trial is actually suing the trial, and this is jeopardizing what's happening in the trial. Next slide, please. Next slide. Now, because the DSMB is just looking at the data multiple times before the trial completes, they would have to worry about potentially inflating the probability of false positives. And this happens when you have multiple looks at the data comparing what's happening between arms, and they would often have a way to actually control for this. Next slide, please. And typically, there's various statistical rules for controlling for type 1 error. And sometimes they would have this as stopping rules or warning rules as part of the DSMB charter or the protocol. And I won't spend too much time on this. I'm just giving you a heads up about one of this. Now, stopping trials early for benefit has challenges. This will be the topic for our next discussion on the 23rd. We'll come to it. But it is important to know that not all trials require a DSMB. Next slide, please. Now, which means if you're involved in a trial, you may want to really determine whether a DSMB is required. Quite often, to determine whether a DSMB is trial, there's 3 criteria that people use in general. One is you really want to look at whether the participants are in a situation where safety concerns may be unusually high. This could be because the outcome is mortality. There's an invasive procedure being looked at. There's worries about toxicity, or you're dealing with a very fragile population of pregnant women, people who are terminally ill or diminished mental capacity, or the elderly. So those are areas where even if you don't have stopping rules, you may actually want to have a DSMB process. And again, I should mention that just having a DSMB doesn't necessarily mean you're going to have a stopping rule. It just means you're just going to have monitor certain things. You also want to make sure that it's practical to have a review by the DSMB. If it's a trial that's recruiting very quickly, the DSMB may not actually have the opportunity to review the data for you to be able to react to what the data is telling you. Also, the DSMB can help to ensure scientific validity or the perception of it. So sometimes you may not have worries about safety or anything, but your worries about this is going to take a long time to do. We want to make sure that we maintain scientific objectivity, and we want this external group of colleagues to help us do that. Next slide, please. All right, in executing this responsibility, so the DSMB, they have the opportunity to look at the data, so they may choose to actually share everything as they go along. Now, of course, the worry about this would be defeats the whole purpose of having a DSMB, because if you're now sharing everything, the concerns would be that recruiting doctors or investigators may actually now start saying, oh, I see what's happening with treatment A, it's actually better than treatment B. I have patients who I really would like to see on treatment A. So you don't want that to happen. So definitely, you don't want them sharing everything about what's happening. Now, you could also say, wait until the end. Don't share anything. Typically, investigators or steering committees are very uncomfortable with not knowing anything at all until the end. So the potential challenge is the idea of sharing with anybody could potentially be problematic in terms of trial integrity and credibility. And I'll touch on some of these issues. Next slide, please. So now I'm going to focus on sharing combined event rate with non-DSMB members. This has become an increasingly common practice. So for the purposes of today's presentation, I'm going to talk about a classical randomized control trial, not an adaptive randomized trial as we've seen during COVID. Many of those trials were deliberately designed to be adaptive. So this is Dave Sackett. And Dave Sackett, for those of you who may not know, he is actually often referred to as the father of evidence-based medicine. He was a colleague at McMaster who studied the Department of Clinical Epidemiology and Biostatistics, the department that I'm in right now. We changed the name to Health Research Methods, Evidence and Impact, but Dave Sackett was the founding chair of that department. Next slide, please. So both of us were serving on a trial where the PI for that trial wanted to actually get this interim combined event rate. And, David and I, as we're talking about it, realized he was not the only one asking for this. Many other investigators have asked for this. So we didn't know what to do. In fact, we had our own views about it. Next slide, please. So what is it? Is when you look at all the events that have occurred in the trial, both in the experimental arm and the control arm, and you combine them into a single number, and you actually divide by the total number of participants that have been recruited up to that time. So it's the total number of patients who have had at least one event divided by the total number of randomized participants. Next slide, please. All right, now the motivation seems to be quite genuine, to be honest, and well-intended. I don't think anybody would want to sabotage their own trial. So they really want to know, are we on track, and are we going to be able to achieve trial objectives? Do we need to do anything to make sure that we salvage what might actually be trouble, and we're not killing patients unnecessarily? Like, really, the motivation for asking for this information seems to be well-intended. Next slide. So, however, Dave used to actually say, this is hilarious. I wonder whether the people really know what they're asking for when they're asking for these things. So he used to describe this request as an hilarious request. Next slide, please. Why? Because the same interim composite event could be compatible with 3 things, all of which could be true, but you just never know which one is true, unless, of course, you serve on the DSMB and you have all the information available to you. If you're an investigator, you don't. All you would have access to is that combined rate, and it could be compatible with exciting news, bad news, or terribly awful news. Next slide, please. So it was unclear to us whether folks were aware such information can be interpreted in several ways, all of which could be correct, or at least one of them could be correct. Next slide, please. Now, consider the following scenario to really appreciate this. So you have an acute illness that kills 30% of its victims, and therefore the control event rate in the absence of intervention is 20%. And you have a drug that could cut this down, 30-day rate, mortality rate, to perhaps by 25%, bring it down to 15% experimental event rate. And with your core investigators, you need about 1,800 participants under these assumptions to be able to actually run the trial to show that it can reduce mortality. So suppose you want to see what's happening after 450 participants. And if you're doing this, you're midway between your control and your intervention, and therefore you're hoping that your event rate is about 15%. And therefore, the control event rate, you would expect it to be around 0.175. Next slide, please. Now suppose your interim rate comes out to be 0.175. That's what you would have hoped for, right? Would you have been satisfied that the trial is on track? Next slide, please. Now, I often would ask people this and ask people to raise their hand. I've never seen everybody raise their hand that they are convinced that the trial is on track. Now, next slide, please. So you could be on target. You're on target because the placebo rate is what you had expected at the beginning, which is 20%. With 25% reduction, you're done it down to 1.5, 0.15 in the experimental thing. And therefore, if you see this, you might actually be excited. Actually, we are on track. So we don't need to do anything, and you feel comfortable. Next slide, please. But you could also get the same number, but that number could actually mean you need more patients. And how could that happen? If maybe the control rate is lower than 20% and the event rate is actually less than what you had actually anticipated. So instead of 15%, it's about 16%. And actually this tells you that, oh, actually you may need to increase the number of participants, but you will never know, right? So only the DSMB would have this information, but as an investigator, you wouldn't know. And suppose now you thought it meant the first interpretation, but this is the interpretation you're in and you just don't know. Next. Or it could also mean that, you know what, the control rate is actually that number and your experimental rate is 0 because nobody's benefiting from this. And that means really this is futile, completely futile. And it's not worth continuing, but you wouldn't know. Next slide, please. Or it could also mean that actually you're killing people. There's more people dying in the control than in the interventional arm, and therefore the numbers are switched. And as a result, there's more people dying in the control arm. Next slide, please. So Dave and I recruited a PhD student to review the literature and survey trialists on current practices to see whether people really have an awareness of these issues. Overall, the majority view was that we wanted to look at what to share with whom, why. There was no clear, coherent message about what investigators running the trials actually thought should be done. What should we be sharing with whom and why? So the review of the literature seems to suggest that really there wasn't a very clear sense of how to approach these issues. Of course, the evidence on these issues was quite low. The quality was quite low. Next slide, please. All right, so the consort people, I'm one of them. And so we actually approached them and said, these are people invested in reporting, transparent reporting of clinical trials. They would actually have a better sense of how to practice. So let's survey them and ask what they thought. Next slide, please. And what we did was we presented them with a scenario similar to the example I presented to you, all right? And we asked them what they thought that information told them. So go to the next slide. So, we asked them, say, this is the rate. What do you think this is telling you? Is it telling you that the drug is performing better than the placebo? Is it telling you that actually there's no difference between them? Or is telling you that actually the drug is performing worse than the placebo? Remember that I showed you that with a single rate, it could be compatible with any of these conclusions. Next slide, please. As it turned out, actually about 50% of them correctly indicated that actually this combined rate can be interpreted in any one of those three ways. About 50% of people were not aware of it, that it could actually be interpreted as such. Next slide, please. Now imagine, think about it. These are people who run trials, they're trialists, they're methodologists, but about 50% of them were really not aware of the challenges of sharing that information. So we conducted an international survey of members of the Society for Clinical Trials and the International Society of Clinical Biostatistics. Eneka and I are regular attendees at the International Society of Clinical Biostatistics. And so we wanted to find out, what do they really think? What do they think we should share with whom and why? Next slide, please. Now, we specifically had different types of information that we asked them whether we should be sharing them. And we paid attention to interim combined rate. How many people do they really think we should be sharing this? And the majority of them said it should be shared with the DSMB, 64% of them. Really? Yeah. And they actually said it should be shared with particularly the steering committee. And their reasons for doing so, or recommending that they should be shared, is that they thought it does not unmask interim results. And it helps this group of people in the steering committee to monitor the trial progress, safety, and design assumptions. Next slide, please. Now, this is the argument they put. It doesn't mask interim result. Is this true? Now, can one use the baseline control rate that you assumed at the beginning of the program to calculate or do some backward calculation of what's happening? Of course, people can do that. They have that information is public in the protocol. You now have the overall combined rate, and you have the control rate that you assume. You can back calculate and try and find out what the actual event rate is in the treatment arm. Now, could you also use other external information on the control rate to try and do the same? The answer is yes. Does this risk unblinding the PI or the steering committee? The answer is yes. There's a potential risk. Next slide, please. Now, it helps to monitor trial progress, safety, and assumption design. It says, really? Then what is the purpose of the DSMB? Shouldn't we actually have trust in the DSMB stewardship? Because we invited them to help us to monitor that. Why don't we trust that they have enough experience, expertise, and desire to help us do that? Why do we still need that? We need to have the DSMB, the steering committee and the PIs also do that in addition to the DSMB. Next slide, please. All right, so we looked at, are there any factors associated with why people think we should share this information? And it turned out that people who were involved in more than 15 clinical trials, some we are, private industry sponsored, actually were less likely to recommend sharing that information because they doubted the usefulness of the information. In other words, people who were experienced in clinical trials, they were the ones who were not in favor of sharing that information. Next slide, please. So in 2018, we organized a special session on this topic in honor of Dave Sackett. Dave Sackett died in 2015. And unfortunately, he died before our student completed the project. And when the project was complete, we invited two colleagues who have been great friends of Dave and have also vested their energy and resources and expertise in training people about DSMB processes. That's Tom Fleming. Go back. That's Tom Fleming and Janet Wittes, and that's Victoria, my student in the middle. Next slide, please. Now, there was some general consensus that sharing with stakeholders can jeopardize confidentiality of the oversight process and the integrity of the results, particularly the interim results, at least during the discussion of the session. Next slide, please. Now, keep going so we can do this. The issue is, what should PIs do if there's general consensus that people are worried about sharing that it could jeopardize confidentiality? I think they should really consider recruiting DSMB members who are very experienced. They have the trial, they're smart, they're wise, and trust them, okay? And they should agree beforehand what rules would actually be used for recommending stopping a trial and how this information will be shared with the PIs to avoid things that could actually lead to unintended consequences. And the PIs, I think we all agree, they should remain blended until the end of the study. So typical DSMB should only involve the PIs when the drug is convincingly beneficial or convincingly harmful, or a change in the protocol is required. Otherwise, involving them outside of this criteria may actually jeopardize that mandate. Next slide, please. All right, there's still a lot of unresolved issues. Should related DSMBs or DSMBs or related trials talk to each other? The answer is yes. Actually, I have served on DSMBs of several related trials specifically for that purpose, where we have one person serving on across all of them to be able to provide those insights to help each other. But this is part of what's in the charter to allow that. Sometimes we've had a single DSMB monitor several related trials for that purpose. I chair DSMB of at least 3 related trials in nephrology for that purpose. Now, should related DSMBs or DSMBs of unrelated trials talk to each other? The answer is no. You jeopardize the risk for confidentiality being breached, so we will not normally recommend that. Should DSMB member of any related trial inform the trials? The answer is yes. This has happened. This could be beneficial because we've relied on insights from other DSMB to actually do that. Now, there need to be some guidelines of when DSMB would actually share information. At the moment, there's actually no clear guidelines of when DSMB could do this. So here's an opportunity for you guys to actually get engaged and see whether it can help the community come up with some guiding principles. Next slide, please. What about other stakeholders? Should we share with the public? We're still talking about interim data. The answer is no. What about participants? No, unless there's a need to change in the consent process, in which case there would be a need to actually then inform participants that you're changing the informed process because there's additional information about risks that were not known before when the trial started. What about regulators? The answer is yes, on the need to know basis. And we've had trials where we've had to communicate with regulators because they wanted to know. Research Ethics Board, also on the need to know basis. What about sponsors? I say it depends. If it's about harms and this could actually be useful, you really might want the sponsor to know very quickly that, hey, you know what? There's some new side effects that we didn't know about. Next slide, please. The most important thing really is for the DSMB charter to be very clear on what will be shared with whom, and the mechanism and the rationale for doing so. Quite often I find most DSMB charts are very, very soft or even silent on these issues. Next slide, please. But I think we would all agree the fewer the people who know, the better for the trial integrity and maintaining that confidentiality. Next slide, please. Now, I'm just bringing it to your awareness that the Society for Clinical Trials actually have a training program for DSMB, and this was led by Dave Demetz, who's also dedicated a lot of his expertise in helping people know about how to best monitor clinical trials. There's a video and there's some slides on the website for that. And we wrote a small article making people aware of these resources. They're available for free. Next slide, please. There's other resources on DSMB. You'll notice that both actually Susan, Tom, and David have been part of many of these efforts. Next slide, please. I just wanted to thank these colleagues because the little I know about this process, I also learned from serving on DSMBs with them, and also from the training that I received from every single one of them. They've been incredible colleagues, and with this, I'll stop. Pass it on back to you, Aneka and Natalie.
Thank you very much. So we have a little bit of time for questions. Actually, we have 10 minutes for questions. Does anybody have any questions? I'll maybe start with one question. Right at the beginning, you mentioned that some trials do not really need a DSMB, and I completely agree. I think we kind of by default think there should be a DSMB. What would the following two things be a reason for you not to have a DSMB? One is a single arm trial. Would that just automatically mean we don't need a DSMB, or do we sometimes need one? And the other one is if it's a not serious condition, like we did a trial on training, getting infants to sleep, for example. Does a trial like that need a DSMB?
All right. So let's start with a single arm trial. Would a single arm trial be enough reason to say I don't need a DSMB? The answer is it depends. I have served on a DSMB of a single arm trial, and the reason why this was put together by the investigators was that they were worried about toxicity, and they didn't have enough information. And a two-arm trial was a non-negotiable issue for the trial. It was a trial in thrombosis, but they said they had some baseline control rate, and they just wanted to make sure that the treatment, which was already approved for something else, really did not have bleeding rate higher than what's already known. So the comparator was, control rate that's already available in the public, not necessarily an active comparator in the trial. So our job was actually just to monitor safety. There were no stopping rules, but we were monitoring, accumulating data on safety, on bleeding, to make sure that it was no different or excessively higher than what was expected based on the literature. Now, what about low risk? I often say it's better to actually go through the criteria for determining the need for DSMB to be able to determine whether you don't need one. So I recommend being very objective in assessing that, not just going by a face value that, oh, we think it's a low risk. So if you go through the criteria, you could actually document that you deliberately, objectively went through those criteria and came to a conclusion that this was a low risk study, and perhaps you may not even have the opportunity to actually review the data to be able to get to it. So I think because we have the criteria, I would rather you go through the criteria.
Thank you. Now, I still don't
Lahana?
see questions. Oh, do
Yeah,
you
we've
have
got
them,
one
Natalie?
we do. We've got one coming through on the Q&A from Myra. I've heard of sponsors who ask if interim results are in the promising zone so they can start to prepare regulatory documents. Is that common or acceptable?
Wow. Yes, I've also been put in that situation. And one would say it is not appropriate unless the charter itself stipulated that this would be done and there's a mechanism of how it will be done safely without jeopardizing the trial integrity and confidentiality. Otherwise, one runs the risk of perhaps not being able to fulfill the mandate of maintaining confidentiality among the DSMB. And actually, this is quite often the case with sponsored industry-sponsored trials that the committee would want to know, or the sponsor would want to know, am I at least in the right direction? They ask those kind of questions. So I think it's dangerous. But if at all something like that were to be contemplated, or rather the discussion be held at the beginning and that be put in the charter and how it will be safely done.
Thank you, Lahana. There's another one that's come through from Francesca. In the context of a low-risk trial, would it still be appropriate or beneficial to establish a DSMB, not necessarily for safety concerns, but to monitor study conduct and data quality?
The short answer is yes. And one of the criteria for doing this would actually be to monitor the trial integrity and scientific validity of that trial without necessarily safety being the main concern or the reason why it was put together.
Thank you, Lahana. And from Chris, great talk. Thanks so much. Your examples focused on binary endpoints. Is there more justification for sharing pooled results with investigators to explore sample size assumptions for other endpoint types? For example, SD for continuous endpoints or event counts for survival endpoints?
The answer is yes, provided this is also in the charter as to the reason why certain information would be shared and who it will be shared with and the purpose for which it will be shared. In adaptive trials, one of the reasons for actually doing interim analysis is specifically to do that, to be able to actually look at whether the assumptions for the design need to be revisited, sample size recalculation and so on. But this is for an adaptive clinical trial. And I think sometimes this could be the reason, but that should be stipulated in the charter.
Thank you. And from Mason, how should investigators approach DSMB recommendations given that they don't have the full information? When is it appropriate to override DSMB recommendations?
All right, it's a very good question, right? Now, so remember one of the things I suggested is recruit people who are really seasoned, who have done this, they know what they're doing, and they would actually be making a recommendation after very, very thoughtful deliberations, and you got to trust them. So I would say, yes, you don't have full information, and it's already known you don't have full information, but you brought them in simply because you wouldn't have full information, but you trust their recommendation because it would be thoughtful. So I think we should really trust the steward. Otherwise, there's no reason for instituting a process for which we don't really trust the stewardship. Now, when would it be appropriate to override the DSMB recommendation? It's just a recommendation. There are times, and I have been involved in trials where we as a DSMB were not even sure what to do. But certainly we thought we might actually recommend a particular action. However, we wanted to huddle with the investigators themselves before we go that direction and say, here's what we are leaning towards, and here are reasons, but we thought we should unblind you guys and now have this discussion with you. but following the unblinding, they will no longer be involved in trial decisions as part of the steering committee. So we would actually go to the steering committee and says, here's where we are. We need to unblind one of you or two of you to be able to have some open discussions. We don't tell them about what, but here are the conditions that if we unblind you, then we'll have an open discussion. You'll share all the information we have and share our recommendation, or at least what we're leaning towards and the reasons why, but show you what our stumbling block is and reason with you about what the best action is. And sometimes that has happened, but this thing should also be stipulated in the charter because there are realities of running clinical trials. And we've also had a situation where you should anticipate that the DSMB people, if you really trust them, if they make a recommendation to do something and you go against it, it becomes a bit of a challenge. So you might want to find a situation, what would we do if we as the principal investigators don't agree with their recommendation? Perhaps a third party could be a way to go. And the reason you're doing that is to actually make sure that you still want to show goodwill, that you trust their stewardship, but you realize that two reasonable people could look at the same information and come to two different conclusions. And usually the way we look at this is just to have a third peer to help us.
Thanks, Lahana. We've just got probably last question from Wesley. Who generally prepares the interim report if the trial statistician is trying to remain neutral and blinded and the DSMB statistician is not resourced to do so?
All right. So typically, we have three types of statisticians in a trial. Is the trial statistician who is part of the steering committee, helps to design the trial and huddle with investigators for problem solving and so on. And then we have a DSMB statistician. That's the person who helps to prepare the reports for the DSMB and to meet with them in closed session so that they understand the data and they could answer all the questions for the DSMB. And the DSMB sometimes could ask them to provide certain information or analysis results beyond what is actually stipulated in the statistical analysis plan. And then you have the third statistician who actually does the analysis for the trial. So, the one who's about the design, the one who's actually operational, does the analysis part, is not really involved in steering committee decisions. And then you have the DSMB support statistician. So typically the DSMB support statistician is the one who prepares the reports. All these things are usually agreed upon right from the beginning.
Thanks, Lahana. Sorry, one just last question squeezed in from Griselda. When setting up a DSMB, would it be better to have members that sit on different DSMBs of related trials, or a single DSMB that can review related trials? I'm thinking about confidentiality and perhaps conflict of interest concerns. Would the recommendation be different for unrelated trials?
Really all depends. I have been in both scenarios where I'm the one DSMB that serves on several DSMB, and this is deliberately done. And I've also been in a situation where I actually chair DSMB of several trials that are related. I've also been in a situation where I'm a member of a DSMB supporting 5 related trials. So it all depends. We don't often have lots of people with this expertise, and you try to make the best use of whoever is willing to help you.
Thank you. Just for the last minute, I know we're over.
I think we're out of time. Yeah.
We're out of time.
Yeah. Thank you so much, and I hope we didn't exacerbate your jet lag. I just want to use 1 minute to do a little bit of marketing for our other events. So ACTA Sting, and Sting is the Statistics in Trials group within ACTA. We're running a DSMB program where we pair mentors with mentees to teach people to be able to serve on a DSMB. You can get the information on our website. Then I just want to advertise our next talk, which is a Sting chat on the 1st of October, where we will talk to the 2 previous winners of the ACTA Sting Excellence in Trial Statistics Award and ask them what makes a good trial. So please diarise that. And also to remind people that registration is open for the ACTA 2025 Clinical Trials and Registry Symposium being done in November. So please register for that as well. And we would appreciate if you could complete the questionnaire, the survey at the end of it. And also remember that there are 2 more talks on DSMBs as part of this series. Thank you for your attendance, and thank you again very much for your time, Leanna.
Thank you.
Thank you so much, Lahana.
Okay, bye-bye for now.
Thanks. Bye.
Thank you.